Hi everyone, this is a lecture on cellular respiration. I'm going to cover the first two stages of a three-stage process of cellular respiration where we use food molecules and convert the energy that's stored in them into ATP. This follows chapter 7.1 through 7.4 of your textbook, which corresponds to these pages. Though personally speaking, I actually don't prefer your textbook, and I actually prefer Khan Academy and some other alternative resources when it comes to learning the three stages of the cellular respiration. At least the info in the textbook isn't bad, but I don't like the images so much. Okay, so just a reminder, in terms of photosynthesis versus cellular respiration, they're not the exact reverse of each other, though there are elements are very similar. For instance, a lot of the reactants in photosynthesis, like CO2 and water, are byproducts of respiration. All right. On the other hand, for photosynthesis, a lot of the byproducts and products of photosynthesis are the reactants for respiration. However, the key thing here is the source of energy or the form of energy that is used or released in both. And in this case, when it comes to energy in photosynthesis, that energy resource is coming from light, while in respiration, the energy that results from it is not light energy, it's ATP, a form of chemical energy. So the process of cellular respiration is, and we call it cellular respiration, not simply respiration, because when we say respiration, we generally, that could refer to the overall process of breathing versus what you do in your cells. So cellular respiration is the process of breaking down glucose into chemical energy and releasing roughly 36 ATP, though that number varies a little bit. Now, in this case, this overall process is what we call exergonic, because we're taking the energy that's stored in a much bigger molecule of glucose, and then we're breaking that molecule into much smaller products of water and CO2. And in addition to that, we're releasing a lot of energy. We're not just releasing ATP, but we're also releasing additional heat, which is often also produced by this process. So when we do cellular respiration, it's the form of making ATP energy for your body, but a similar process happens when you burn wood, and in this or any other food molecule you can actually burn through a process called combustion. You don't really need to know combustion, but it's the similar idea is when you burn wood, you're taking a carbohydrate, and you're burning it and you're releasing energy. But in this form of combustion, because you don't have any bio stuff around it to absorb the energy, all you release is heat. But in respiration, it's a much more controlled and multi-step process. In this case, instead of converting all that energy to heat, we're converting a significant chunk of it into ATP and capturing its energy in our cells. And just a reminder, we're going to use these terms a couple times, but you need to know when a product is oxidized versus when a product is a result of reduction. So generally speaking, when you're building up a molecule and making it larger or storing energy into it, that's an endergonic process. And when you're breaking a large molecule down and making it smaller and releasing energy, that's an exergonic process. Now, a lot of the times when the overall product becomes exergonic, for instance, or releasing energy, there's also another thing becoming endergonic, meaning another thing absorbing that energy. So for example, when glucose is undergoing an exergonic process to release its energy, it's thus adding energy into ATP, which is undergoing an endergonic process. Now that may be a little bit confusing, but I'll just know I'll point it out when we go throughout the lesson, when something's undergoing exergonic reaction versus an endergonic reaction. Now, another thing here is we learned about energy carriers molecules in photosynthesis. You learned about ATP's use and the use of NADPH. Now, in this lesson, we're going to also use ATP again, but our electron carriers, so the form of carrying energy via molecules that hold special highly energized electrons, these carriers are different. Now this time our full battery, our highly charged molecule, is NADH and FADH2. And the empty batteries are NAD plus and FAD. So these are our empty batteries. So when we have this, what we want to do is get energy and store it into it. And when we have FADH2 and NADH, these are our fully charged batteries, and we get to use those energy batteries to do other stuff to it, generally convert it to ATP. Now be careful, because NADH and NADPH is used in photosynthesis, and it's very easy to mix them up. So a real easy way to remember is NADPH. While the whole name, it stands for something else, I like to think, hey, the P is for photosynthesis. While the lack of the P in NADH means it's used for something other than photosynthesis. In this case, it's the battery used for respiration. And when I say battery, I mean the molecule we're using to charge up and hold energy that we can utilize elsewhere in the process. So whenever we charge our batteries, we're going to reduce them, right? We're charging it. And whenever we break down the batteries and turn it to NAD plus or FAD, we're going to oxidize or release and use that energy. Okay, so the three main stages of cellular respiration is glycolysis. And glycolysis is the one process in cellular respiration that doesn't require oxygen and doesn't need a mitochondria. Earliest life on Earth did only glycolysis. And then until they were able to obtain a mitochondria, and then they were able to do the more complicated processes of pyruvate oxidation and Krebs cycle and ETC and oxidative phosphorylation. Sometimes this pyruvate oxidation is also known as acetyl-CoA formation. And I'll refer it to mostly acetyl-CoA, but either form of the word works. Now, these two processes require oxygen, and they require the presence of a mitochondria. So if you think about it in terms of the kingdoms of organisms, all organisms can do glycolysis, including bacteria and what is the other one? Archaea. But all the other eukarya kingdoms can do aerobic respiration. And by the way, this is the overall equation you should know for respiration. And the energy range is generally somewhere between 34 to 38 ATP is released through respiration. There's a little bit of variance based on the different type of tissue the respiration is occurring in the body. Now, the big picture for respiration. The first two stages are actually, they release very little energy, or they capture very little energy. You get a little bit of ATP from the first two stages. But the real meat of the first two stages, at least for larger organisms that need a lot of energy, is the creation of the batteries, the creation of NADPH, or not NADPH, sorry, the creation of NADH and FADH. And in the very last step, all those batteries of NADH and FADH that you've collected from the first two steps is completely converted to ATP energy. And that's where the meat of the reaction is. Now, if you're a small organism like a bacteria or archaea, you can get away with only making a little bit of energy from the sugar. But if you're a large organism, you want to get as much energy out of it as possible because you have a lot more metabolic needs, and that means you need to make the maximum amount of energy possible. Now, we're going to go over glycolysis. This is the first stage of respiration. So glycolysis is an anaerobic process, and again, it's the simplest way of releasing just a teeny bit of energy. And it doesn't require any special equipment. It's just simply catalyzed by enzymes in your cytoplasm, and all organisms on Earth can do glycolysis. So in this case, we're going to take sugar or glucose, and we're going to convert it to a smaller sugar, or not sugar, sorry, a smaller 3-carbon molecule called pyruvate. So we're going to crack it in half. And in this process, we're going to release a little bit of energy. Now, it's a teeny bit confusing, but what you need to do is initially in the process, you're going to use up 2 ATP to kickstart the reaction. And then there's a bunch of little sidestep reactions that you have to do. But ultimately, those sidestep reactions between glucose to pyruvate are going to end up resulting in 4 ATP released, and it's not showing that in this picture, but it's 4, and 2 NADPH released. Now, the reason why this picture might look a little bit confusing to you is because for each of the 2 molecules, or 2 pyruvate molecules that are going to be made from glucose, each one of them is going to ultimately result in the release of 1 NADH. and 2 ATP. Now, I'm not a huge fan of this diagram. It's decent, but I think for your purposes, I'm going to draw a much simpler form of this diagram. So in this case, we have glucose, we end in pyruvate, and overall in this process, we're going to invest 2 ATP. We do a little bit of endergonic investment. And then ultimately, though, we're going to get a lot more energy than we invested, and we're going to get 4 ATP from this result. And that's going to result in a net overall earning of 2. Like we spent $2, we earned $4. Overall, we gained $2. And in addition to that, we get 2 NADH, which we're going to be able to turn into a lot more ATP. So in summary, in this reaction, we get 2 ATP, and we use 2 ATP and we burn those up, and then we get 2 ADP empty batteries. But then, as a result of that, we form 4 ATP from this initial step that leads us to the release of 4 ATP, and we release NADH, which we get to convert to ATP later on. This is the basic diagram for glycolysis that we usually teach in BioH. However, if we're only asking you on exam to summarize the overall reaction, then what you actually really only need to know is that 2 ATP are formed from this process, and 2 NADH are formed from the process of converting a 6-carbon glucose into 2 separate 3-carbon pyruvate. And remember, this all happens in the cytoplasm, and it does not need oxygen. Okay? Now, this process and the process of forming ATP in the second stage both don't end up with all that much ATP. They only form 2 ATP, and that's because the form of producing ATP is simply done through enzyme catalyzed reactions, which just help smush the phosphates onto the ADP molecule to form ATP. And it doesn't do a fabulous job of it. It takes a lot of, it's not efficient. The form of making ATP using an ATP synthase molecule and a hydrogen gradient, that's actually way more efficient, and that's not used in this process. So this process of crappy ATP making we call substrate level phosphorylation, because it's using a substrate it's using a molecule as its substrate to force the creation of ATP, the phosphorylation of ATP using an enzyme. So the advantage here is you don't need as much equipment, and it's much faster, but it's a lot worse at making ATP compared to the way ATP was made, for instance, in LDR, and the way it's going to be made in stage 3. But it is the main form that ATP is released in the first 2 stages of respiration, in Krebs, which is the second stage, and glycolysis, which is the first stage. Now, just a reminder in terms of the words we use, right? Glucose is losing energy and releasing it, so it's being oxidized. But notice, ATP and NADH are charged up. They're the higher energy forms of the molecules. So those guys are being reduced. Okay, so just always track that. Even though a process is oxidized, there's always something else being reduced, right? There's that coupling of the reaction. One gains energy, somewhere else is losing energy, and vice versa. All right, now this is just a start. Now we have pyruvate, and if oxygen is present, and if we're in an organism that has a mitochondria, we can go further and process and get more ATP from all of this. If you're a bacteria or archaea, you're out of luck. Those 2 ATP formed from glycolysis is all you get in terms of energy from glucose. But if you're a eukaryote, you're going to be able to get at least 34 more ATP out of it. So we call this the branching point. So if oxygen isn't present, you can't use your mitochondria well, and you're going to do a process called fermentation, also known as anaerobic respiration. Anaerobic meaning there's a lack of oxygen. Aerobic means use of oxygen. You do aerobic exercise, you're burning up a lot of energy, and you need a lot of oxygen. But in this case here, if we do have oxygen present and a mitochondria, because we're eukaryotes, then you're going to be able to go to the next stages of respiration, also known as aerobic respiration. And the second stage of respiration that we can now enter with our pyruvate is stage 2, which is partially the Krebs cycle and an initial pre-step before the Krebs cycle. Okay, so before we can do the next 2 stages of respiration, we need a mitochondria, and we need to have oxygen present. Now, the second stage doesn't directly use oxygen, but it relies on the presence of oxygen in order to do its job. And I'll explain why later in either this lecture or the next lecture. Now, before we do this, we need to understand the structure of a mitochondria. So, the mitochondria looks like this, and I do encourage that you kind of draw something like this out. So, it has an outer membrane, it has an inner membrane, and that inner membrane has a lot of extra folds to make it have a lot of surface area, because the inner membrane is the meat of the Krebs cycle and the third stage, oxidative phosphorylation, because it has a lot of essential enzymes, especially needed for stage 3. So we need to add lots and lots and lots of membrane proteins. So we need as much membrane available as possible. Those individual folds of the membrane in the inner membrane are known as cristae. Now, between the inner membrane and the outer membrane is the space, and I'm just going to color in a little bit. So that gap between the inner membrane and outer membrane is a lot small, is pretty thin, and that's actually really important because that's a zone where we pump hydrogen into to form a hydrogen gradient. So that's known as the intermembrane space. Inter, like intermediate, means between two things, so intermembrane means between the two membranes. And finally, the last space or chunk of the membrane that we should know is the matrix, and the matrix is the very inside middle of the mitochondria, and that's actually where stage 2 happens, the stage after glycolysis, and that's the inside space. So we have our pyruvate on the outside of our mitochondria, and to get to stage 2, we're going to need to shove this pyruvate all the way inside the matrix in order to do this stage well. And this is just a summary of the mitochondria structure and all its stuff, which I just stated earlier, with its inner membrane, with its folds and cristae, as well as that intermembrane space, and why they're important. So you should know how each of these spaces in the mitochondria are useful for the different stages of respiration. And again, the inner membrane is really critical for having an electron transport chain and ATP synthase, which you're going to need for the very final stage, which is super important for converting all those batteries, NADH and FADH2, into ATP. So what we're going to do is now in the inside, instead of the inner membrane, we're going to take that pyruvate and crank out as much of the batteries, NADH and FADH, that we can make so that those batteries can then be used in the membrane, inner membrane, to convert it into ATP. So, stage 2 of respiration is broken down into 2 stages. The first stage is the stage where pyruvate converts to something else called acetyl-CoA formation. And that process of it turning to acetyl-CoA formation is also going to help that pyruvate move into the inside middle of the mitochondrial matrix. So it's called acetyl-CoA formation because a molecule called acetyl-CoA is formed. It's also known as pyruvate oxidation because pyruvate is being broken up, right? Oxidation is loss. So pyruvate is losing energy and becoming smaller because it's becoming smaller when it becomes acetyl-CoA. Because pyruvate has 3 carbons while acetyl-CoA has 2 carbons. It's also going to gain a coenzyme A, and that coenzyme A is going to attach to the molecule, and that's going to actually help it enter the next stage of this process. Okay? In addition, you're going to lose one of your carbons, so you turn from a 3-carbon molecule to a 2-carbon molecule, and again, very critically, you're going to gain a little bit of NADH. And that's the main process here. So that's a lot of information to remember. So if you don't need to memorize the amounts, very helpfully, you just want to remember each pyruvate as it enters becomes acetyl-CoA, which only has 2 carbons. That coenzyme A had to come in somewhere, so it's going to come in. And because we lost 1 carbon, a carbon dioxide has to leave. And a little bit of energy needs to be released. And in this case, we're going to get some NADH. So this is the overall reaction of acetyl-CoA formation that you should be able to track. And this process is also going to help it move into the inside of the mitochondria matrix. Now, this process sometimes utilizes a little bit of energy, so that accounts for the range of ATP that's formed, because you make 32 to 38 ATP overall from this breakdown process, sometimes even 40 if it's very efficient. And the reason why is sometimes the speed at which you need to transfer this acetyl-CoA and convert pyruvate and transfer it to acetyl-CoA and move it into the matrix is what ends up accounting for slight differences in different tissues in your bodies, making a lot of ATP. Some of them make it a little bit more on the higher end, like 40 ATP. Some of them make it on the lower end, like 32 ATP overall, because they're wasting a little bit in this process. But you should understand that this is the overall end result of the first step of stage 2. Now that we're in the matrix, we can enter the next step of stage 2, which is the Krebs cycle, which is this really long, very complicated process, not unlike the complicatedness that is involved in the Calvin cycle. The Krebs cycle is also very complicated in that there's many, many steps with many, many enzymes involved, and it's vastly simplified in our class. So in this case here, our acetyl-CoA joins the cycle and it combines with oxaloacetate. So if you remember the Calvin cycle, CO2 comes in and is combined with RUBP. In this case, acetyl-CoA comes in and is combined with oxaloacetate. It briefly becomes a 6-carbon molecule called citrate that accounts for Krebs cycle's second name, which is the citric acid cycle. Krebs cycle is named after the guy who discovered it, Krebs. And then it undergoes a bunch of sub-processes that eventually release a lot of NADH and a little bit of ATP and a decent amount of FADH. But the main thing it's releasing is a good chunk of NADH. In this process, the acetyl-CoA is completely broken down, and those carbons of acetyl-CoA are going to be converted to CO2, so those are going to be lost. And then you get released, and the oxaloacetate that started it all is regenerated, and it can continue taking in acetyl-CoA and regenerating. Now, this is a lot of information, so if I were to ask you to just kind of summarize the overall consequence of the citric acid cycle or Krebs cycle, I would just simply draw this. So, acetyl-CoA, the 2-carbon molecule comes in, and the coenzyme A that just helped it shove into this cycle gets to leave. And then in this process, a bunch of things happen. CO2 gets to be released, so a bunch of CO2 leaves. So 2 CO2 actually leave, so 1 CO2, a second CO2. A little bit of NADH is made, 3 NADH in fact, from 3 NAD+. And 1 FADH is made. Okay? And 1 ATP is made. And that's basically what you need to know. And in some cases, like on our final, I won't even ask you to memorize the number. You just need to know acetyl-CoA comes in, the coenzyme A leaves, all the carbons leave as carbon dioxide, you get NADH, FADH2, and ATP. And that's all you really need to know for a final, for example, where I don't ask you to know the numbers. So you just need to know at the end of the stage 2, you get the acetyl-CoA releases all its carbons as CO2, and you get NADH, FADH, and ATP energy molecules. So now at this point, our molecule of glucose has been completely oxidized, aka broken down and energy lost, and turned into CO2. But we only have 4 ATP. We need to make the rest of the ATP, and that's where all those batteries we just formed we're going to use. And remember, we had all that NADH and FADH in the middle of the matrix. We also, the NADH that was outside in the cytosol or cytoplasm is going to also be transported eventually to that matrix. So we have a nice big clump and leftover NADH and FADH all nicely in the middle of our mitochondria. And now we are ready for the next part, which is known as oxidative phosphorylation, or stage 3. And all those electron carriers of NADH and FADH2 are going to release energy and be used to make a concentration gradient that will allow us to use ATP synthase and churn out a ton of ATP. And that is the true value of the Krebs cycle, and that's actually the true value of all these earlier stages of respiration in the in the creation of all these energy carriers that we can actually convert to just raw ATP and a little bit of heat, but a lot of ATP as well. So in the Krebs cycle, we have reduced it all into NADH and FADH2. Those have been reduced because they have been charged up and gained energy, while our acetyl-CoA has been oxidized, also, which was pyruvate eventually, or ultimately. So pyruvate oxidizes into acetyl-CoA, acetyl-CoA oxidizes and is used to charge up these batteries. And then in the last stage, oxidative phosphorylation, we're going to oxidize all that energy from our energy carrier batteries, NADH and FADH, and convert that into ATP. And that's the whole point. We need that ATP to run our bodies, to move our muscles, to build molecules. We need it. And that's why we need lots of cellular respiration. Okay, so this is kind of a helpful kind of summary of the 3 stages of respiration, and feel free to freeze the frame on here and write everything down. You also have access to the slides. But remember, stage 1 and 2, the whole idea is you get a little bit of ATP, but you're truly setting the stage for the final stage where you take all those energy carriers and harvest the most ATP. Now you'll notice the last stage didn't use oxygen, but it won't happen without oxygen, and I'll explain why in the next lecture when we go over stage 3. But there's a reason why stage 2 cannot happen without oxygen being present. And it has everything to do with the reactants that are needed for it. So, like I said, this is a really helpful kind of summary of everything. And I really do encourage you to watch Crash Course. I think the video they have on respiration is pretty effective. And I also, if you need to look up and read more on respiration, I actually recommend Khan Academy's resource on cellular respiration, as well as the resources on photosynthesis, because I think they're really effective. And they have good diagrams. So these are the takeaways. Just basically, a little bit of ATP is made in the first 2 stages of cellular respiration, an exergonic process for releasing energy from glucose. That energy is originally formed through substrate level phosphorylation, which gets you a little bit of crappy ATP. Like you get ATP itself isn't crappy, but you get a crappy amount of it. But what's really happening is with glycolysis and stage 2, you ultimately get to form a lot of these energy carriers of NADH and FADH2, and those will ultimately be able to be converted into ATP. And you should know where each of those stages occur and what their overall products and reactants are. And these were the key vocabulary that was introduced, and you really need to know the anatomy of the mitochondria and what occurs where in the mitochondria. All right, good luck. Bye.